Now, let me show you again just an illustration. There are three colors that you should be, well, four actually, that you should be familiar with that you're going to see throughout your textbook. In the differentiation process, we concern ourselves primarily with three main colors: blue, red, and yellow. So what do these colors represent? Well, blue is typically used to represent what we call the ectoderm. Okay, so these are the germ layers. When a zygote, which is the fertilized egg, starts dividing and becoming multiple cells and undergoes morphogenesis, or the movement of those cells around, starts forming into these various layers, what we call germ layers. And each germ layer has a particular fate or derivative, or I should say, set of cells and tissues that it will become. Due to the nature of where it's found, that's its fate, so to speak. So the ectoderm, and each organism can have a different assortment and arrangement of these. So when you look at cells and when you look at these embryos throughout your book, blue is the universal color that we give to the ectoderm. Mesoderm is red, and all of the mesoderm components that come from that are in red. And yellow is used to illustrate the endoderm. Okay? Now, purple is not used as often because it doesn't always show up in the initial stages, but that's the germline cells. Those are the sex cells or the stem cells that will form the gametes in an organism. Most of the time, we don't really get to that point in most of the pictures to show the purple. So mainly it's going to be blue, red, and yellow. In fact, let me show you a cross-section of a frog or xenopus embryo. This is kind of what I'm talking about, where the outer layer here in blue, this is going to become things such as the skin, as well as the nervous system, the brain and the spinal column and the neurons. Now, that is one thing I'm going to test you on, just your basic understanding of what the derivatives of each germ layer are. Because that's really most of what we're going to study this entire semester is how does the ectoderm become the skin and become the brain and become the spinal column and whatnot. How does the mesoderm become all of its components? Okay? So, again, there's three main things that the ectoderm becomes. It's essentially the skin. It becomes the nervous system. You could split hairs and say it also becomes the pigment cells of the skin, but that's part of the skin as well. So the two main derivatives are skin and neurons. So skin and neurons are derived from the ectoderm. Mesoderm has quite a bit more to it. Mesoderm has a lot of stuff. For example, it becomes your bone. It becomes your muscles. It becomes, your kidneys. It becomes your gonads, or the sex organs. It forms certain parts of your spinal column, but not neurons. More of the connective tissue in between the vertebrae. But again, that's part of bone and cartilage. So mesoderm, its main components are bone, muscle, kidneys, and then the sex organs like the gonads. And then the endoderm is the two other main aspects of physiology, which are your digestive system as well as your respiratory system, your lungs and things of that sort. So all of the components of your digestive system, your esophagus, your stomach, your liver, your pancreas, your large and small intestines, all of those are derived from the endoderm. So the endoderm, again, now, one of the things that confuses people is don't think that endoderm just means inside, because, in fact, the ectoderm becomes your nervous system, which is one of the most central parts of your body that's protected by the other layers. So just because we say ectoderm and skin is one of those derivatives, that's not just because it's not just an outer layer. Okay? Another thing that people tend to confuse is the terminology ectoderm and epithelial. So let me explain the difference between those two. Ectoderm is a germ layer. However, all three of these germ layers can form epithelial cells. So what's the difference? Epithelial cells are typically a covering of sorts. So even your esophagus, which is part of your digestive system, has epithelial cells that coat and that protect the, as your food goes down and so on and so forth. So epithelial cells just refers more to the nature of the cells being a covering and a coating, okay, rather than it being synonymous with ectoderm and skin. That's one of the things that people tend to confuse. Morphogenesis is the process of the cells changing their shape and their place during development. Differentiation, on the other hand, is the process of a cell having a different function. Okay? So that's one of the main differences between the two. I'm going to explain things a couple of different ways just so it hopefully sinks in. So again, differentiation. Again, think about it in terms of you going to school. The further along your path you go, the more concrete or set you are in that discipline. And the same thing is true for cells. And that's one of the things that we study in development and its application, because can we take cells that have gone down this path and turn them into something else? Well, yeah, we can. It's a very difficult process, but we can take skin and turn it into neurons. Now, hopefully, you know that it's not that big of a step to do so because they both originate from the same germ layer. So it's not too difficult to take skin and turn it into neurons. It is a little more difficult to take skin and maybe turn it into something else, like muscle or some type of endoderm cell, because they go down a very different path in their differentiation. All right. Now, in the morphogenesis process, where the cells change their position and their shape, I said that there were two different forms in which the cells can be found. You have the epithelial cells, and then you have the mesenchymal, or sometimes it's called mesenchyme, cells. So epithelial cells essentially are connected in one form or another to other cells. Now, the way in which they're connected can vary. There's quite a number of ways in which they can form certain structures. Mesenchyme cells pretty much are easy. They're not connected to other cells. So, and cells can go from epithelial to mesenchyme back to epithelial during this process of morphogenesis. They don't always stay as one. They can originate as epithelial, then they change and they become mesenchyme as they move, and then they'll reform new sheets and become epithelial cells again. So really, again, epithelial just means connected, and mesenchyme means loose or not connected to other cells. So again, there's a wide variety of ways. One of the words that I will use quite a bit, so just be familiar with it for later on, is delamination. Because delamination is one of those processes, one of the more common processes when cells go from an epithelial to a mesenchyme transition, where they go from a sheet of cells to individual migrating cells, so to speak. Mesenchyme cells, again, there's other terms that we'll use. Most of these you're not going to necessarily see, but condensation is one of those. Just like water vapor condenses and becomes water droplets, condensation is a transition of cells from a mesenchyme to an epithelial state. So they're literally aggregating or coming together and forming a ball or forming a sheet or whatever the case may be. So delamination and condensation, those are two that I use more often than any others to describe these transitions from epithelial to mesenchyme and back again. Okay, so delamination, that's just one that talks about cells going from an epithelial to mesenchyme. And condensation is one where the free-floating mesenchyme cells coalesce together and form an epithelial connective type of tissue. These are part of morphogenesis. Okay? They're not necessarily used to describe differentiation. Okay? I really am emphasizing this. I know I'm kind of overkilling it, but this is one of those things that even by the end of the semester some people have a big problem with. So morphogenesis, the changing of the shape of the cells as well as their position, that's epithelial and mesenchyme cells. What shape are they? What cells are they associated with? Where do they move to? Where are they migrating? That's all morphogenesis. Differentiation comes down to what genes are being expressed and what proteins are being made in the cell. That's where differentiation comes down to, is what genes are expressed and therefore what proteins are being made. That's the cell's function.